Hello everyone, my name is Malcolm Todd and I'm a grad student with the University of Saskatchewan. Thank you for joining me today for my presentation on exploring genetic variation using basic variation graphs. To begin today, I'll start with a topic introduction, discussing several of the key components of this work. The first of these is structural variants, which are large-scale genetic variations that are classified based on the nature of the rearrangement that occurs, including deletions, insertions, duplications, inversions, and transpositions. Now, traditionally, SVs have been found using sequence alignment. However, many of these methods have often been shown to lack sensitivity, which necessitates our need to go and explore alternatives for how they might be detected. This leads us to the second major component of this work, which are genome graphs that embed multiple genetic sequences in a single graph, allowing us to compare and contrast the different relationships between them. Now, in modeling sequence data, there are two common graph styles. The first of these are K-mer graphs, which decompose the larger sequence into a series of fixed-length subwords that overlap. And the second are string graphs, which decompose the larger sequence into a series of variable-length vertices containing different length subsequences. In this work, we conducted several experiments using small simulated sequences shown here to observe the different patterns that these SVs create under different graph styles. During this work, we explored 4 different graph construction methods to compare the graphs they created when modeling different types of variation. The first of these was a K-mer graph, which was constructed by dividing the sequence into a series of fixed-length subsequences, starting at each character, and then adding edges between any adjacent K-mers that occurred in the sequence. Additionally, we constructed 3 styles of string graphs since they offered us flexibility in how the sequence could be divided. The first of these was a suffix tree-based string graph that located the longest repeated substrings, which served as our vertex candidates. The second was a string graph divided into candidates based on the local minima locations in the longest common prefix array, which was derived from a Burrows-Wheeler transform of the sequences, similar to the approach used by Preza et al. In 2020 for detecting short variations. Finally, a manual approach was taken to construct an optimal string graph that minimized the number of vertices required to model the sequences, while also providing clear boundaries around each vertex location. Additionally, the paths in the Burrows-Wheeler and optimal string graphs were also restricted to prevent any cycles from occurring within the paths to explore the reduction in path complexity that this creates. So here we have the resulting graphs produced, which demonstrated SV patterns consistent with those documented in the literature. These patterns include bubbles at inversion and deletion locations, cycles at duplication locations across the repeated subsequences, and differences in the numbered edges when comparing paths that were transposed versus those that maintain the original ordering. Concerning the strengths and weaknesses of each graph, the K-mer graphs provided clear SV boundaries around each variant location, but required many vertices in order to fully model these sequences, making them rather complex. By contrast, the string graphs required fewer vertices, which made them less complex. However, they could not guarantee the optimal division of the larger sequence. For example, the suffix tree-based graphs failed to guarantee that the SV boundaries would occur between vertices, which could complicate SV identification if we used these graphs. Conversely, the Burrows-Wheeler string graphs provided clear boundaries around variations, but they did not produce a consistent division of the subsequence outside of these variant locations, often introducing redundant vertices, which suggests that further work is required to refine this approach. Restricting the string graphs to prevent cycles in their paths did remove the ambiguity in the graph's paths caused by repeated subsequences. However, the restriction also complicated the process required to calculate the total number of copies of each repeated subsequence in the sequences being modeled, which is a necessary step when locating duplications. Therefore, further research will be necessary to uncover how to locate duplications without introducing this ambiguity into graph paths. So to conclude, this work represents a valuable introductory step for exploring variation using variation graphs and serve to identify several future research directions and necessary refinements requiring further exploration. I want to thank everyone for watching our poster presentation today, and for any additional details, please consult the attached poster PDF or feel free to reach out at the email provided at malcolm.todd@ucesc.ca.